Instead of spending a long time on Thursday going over details for the lab, I'm going to do a little bit of a pre-lab lecture now. There might be some few announcements that I'll make at the beginning of lab, but for the most part, the material that you need to understand the lab will be covered in this short video. So when you enter lab, you'll notice a cart that contains gloves and some masks. Go ahead and grab gloves. Make sure you grab the gloves first so that you are gloved up when you reach into this bag for a mask. So grab a glove, put them on, and then you can grab a mask. And there are different sizes. If any of these are out, please let me know. Then you will find an open seat that has this seat available sticker on it and all of the stuff that you need for lab. If you're in the afternoon lab, some of the material will be found in your drawer instead, and we will go ahead and label the drawers. You can pick the one that you want and put your name on it with tape that is on the bench. So go ahead and label your drawer. That will be your drawer for the duration of the semester. So if we look at some of the material that is at your bench, the first task that you need to do is to calibrate pipettes. You will practice using all 3 of the pipettes that are at your station, but you only need to worry about calibrating one. And so the largest pipette, the one with the blue push lever, you will use these tubes that are in the container. They will hold over a milliliter. They'll hold 1.5 milliliters. For these other tubes, you can practice pipetting into tubes that are found in this baggie. And these baggies are labeled with a seat number as well as AM, PM, and whether you're group A or B. So you'll use this colored water to practice pipetting. You can use these tips. The blue tips go with the blue lever. The yellow tips go with this orangish lever, and the yellow lever you can use either the smaller purple tips or the yellow tips. The purple tips hold a smaller amount of volume. You can kind of control it a little bit better, but they will not push off with the release. The yellow will push off with the release, but might not have as much control. And then you either have a large beaker or a coffee canister that you can dispense of your tips in when you are finished with them. So there are 3 analytical balances throughout the room, so we will have to share them as lab progresses. So just kind of be aware when that's open after you're done practicing pipetting. You can use those analytical balances. And there will be weigh boats that you can use to weigh your water. And each time you weigh your water, you can tear the balance and put another drop on. You don't have to remove the water, you just would tear it each time to take those measurements. And I changed the lab that you only need to take 5 measurements instead of 10, just so that we can cycle through the 3 balances a little quicker. So that's the pipette calibration. So you might need to get started on the column while you are waiting for the analytical balances, because loading and running the column may take some time. So let's talk about the principle behind column purification. This is a way that proteins are often purified. Other things can be purified as well, but it's mainly for proteins. And so this is a form of chromatography. You have probably done TLC or thin layer chromatography in organic. Column purification, or the specific one we are using is size exclusion chromatography, is another type of chromatography that's commonly used in biochemistry. And so what happens, these different columns are over time. And so what happens, you pump your sample on. In our case, we'll just be loading it with a pipette. We don't have a pump that we're loading it on. But the sample gets loaded. And then over time, the sample gets separated. And if there are multiple proteins in that sample, which in our case there will be, those different proteins will get separated depending on the relationship between the column and the buffer that is also flowing through the column. So we have that stationary phase, which is the column itself, and the mobile phase, which is the buffer that goes through. We'll talk about in class a lot of different types of column chromatography, but in this case we're using size exclusion chromatography, which separates proteins based on size. And so again, this is over time. Over time, we have this mixture of proteins that will get loaded, and the large proteins will come through the column first, and the smaller proteins get held up in the column. The reason for this is the column is made of these beads that contain pores, and the small proteins can get stuck in the pores and held up. So they have a longer path to really get through until they get to the end of the column. Larger proteins do not get caught up in these pores and can just go straight through. They have a very short path length to get through the whole column. So they will be removed first. And so for your experiment, we'll be separating 3 proteins of known size so that you can kind of calibrate your column. And then we will run an unknown protein through as well so that you can estimate what size that protein is. So for your experiment, the first thing you need to do is make the column. You need to actually pour the column. And so this is your column. It contains the column itself, a reservoir that will hold buffer. It contains a porous disc so that the beads will not come through. And it contains this cap that will close it. Almost like when you're doing titrations and you have a burette and you can close it off, we just do that manually with a cap. I would highly recommend practicing removing this cap before you put anything into the column. So this cap is connected to this lower fitting, and it's really easy for this whole thing to come off when you're trying to just remove the cap. If that happens, all the work that was in your column is now gone. So before you load anything, I would practice removing just the cap, just the blue piece. And so when you pack your column, you'll go ahead and tape your column up to the ring stand. Either one of these or the actual ring stand itself is fine. I would not recommend clamping it on. This is a pretty thin plastic and it could clamp the plastic, so I would recommend just taping it. This small beaker is your hydrated beads. These are those porous beads. There's a lot of buffer in them. I would recommend mixing this up so you can just swirl it around and pouring it onto the column. And you'll pour a little bit at a time until eventually the hydrated beads will end near the top of your column. The lab gives you a certain measurement. You can measure that. It can be an estimation though as well. You have extra column buffer to help you load that. And while you're loading it, the buffer will be flowing through at the beginning. You eventually need to close it so that you can add more, but buffer will flow through so that you don't overflow your reservoir. While the buffer is flowing through, you will collect it in this collection tube. And this can be reused. So if you run out of buffer, you can reuse this collection buffer because it should all be buffer. So once you have packed your column, you'll then load the column with your knowns. The knowns are phenol red, myoglobin, and blue dextran. You will find a sample of the myoglobin in the freezer along with the unknown. And the myoglobin and blue dextran are already mixed for you and already separated out. You will just add the phenol red to that tube. And then you will load that using a pipette onto the top of your column. Throughout all of this, you want to make sure that you never let the column dry out. So even when you're loading the knowns, you want to make sure you continually have buffer on top. You do want to be careful that your knowns get onto the column before you add extra buffers so they don't get diluted. As soon as your knowns are loaded into the column and they start moving through, you want to be collecting everything into this graduated tube. So the tube has some graduations on the side so that you can read the volume. Again, as soon as the knowns get into the top of the column, you want to start collecting with this tube. Then, as each color comes through, at the point of the strongest color, there will be 3 colors: red, brown, or yellowish, and blue. You'll record the total volume in the graduated cylinder. So each volume will be higher because you've kept that volume in at all times. Then you'll do the same thing with your unknown. You have an unknown tube, you'll add phenol red to it and perform the same thing. You can dump out this graduated tube before you do that, and you can just dump that into the sink. So then you'll repeat the total volume. Note that the phenol red should be the same volume each time. If you do this carefully, you should get a very similar volume. If not, you'll need to adjust the volumes, add or subtract, so that the phenol red is the same as before. Then to perform the analysis, I've given you the known molecular weights for these known samples. For blue dextran, it's actually a very large molecular weight, but you will use only 100,000. This is grams per mole or Dalton, because the void volume of the column, this is the maximum that it can read. So it will never get to 2 million. So anything 100,000 or larger will read as 100,000. And then you either have cow hemoglobin or cow blood or rabbit hemoglobin, rabbit blood. And so you will record the volume that those come out. This is your unknown sample. And you'll, in Excel, plot the elution volume versus the molecular weight on the y-axis. And this will need to be in a log scale. So I can help you do that in Excel. Maybe I'll do a short video on how to do that at some point. So you'll make a plot. It should be a straight line, and it should be a negative slope like this. And you'll find your unknown and then determine, based on the equation for the line, what molecular weight that was. And I will provide you what these look like so you can compare what it should be. So that's a little pre-lab lecture for this lab, column purification, as well as the pipette calibration. The lab report will be due next week, and you will be virtual next week. So I hope this was helpful, and I will see you this week.